So in this study, we wanted to test how different CSF biomarkers of synaptic function, which are neurogranin, SNAP25, synaptotacmin 1, and GAP43, how they associate with brain changes in terms of structure and function, specifically in individuals that are cognitively unimpaired, but at higher risk to develop Alzheimer's disease. So what we did is voxel-wise analysis to test these associations in the whole sample, and then we were interested in knowing whether these associations were modified by AT status, so by Alzheimer's disease pathology. And we found that in the whole sample, the 4 synaptic biomarkers were associated with, so higher levels of these synaptic biomarkers associated with higher brain metabolism and also higher gray matter volume. However, these associations were modified indeed by AD pathology in the sense that for brain metabolism, the higher levels of these synaptic biomarkers associated with increased brain metabolism in those participants with amyloid and tau pathologies. While for gray matter volume, we found a different pattern in which in this group of amyloid and tau positivity participants, they showed lower or negative association with gray matter volume. Well, the results, I think it's like a first result showing that what these synaptic biomarkers reflect in terms of brain pathology in the very early stages of Alzheimer's disease in cognitively unimpaired participants, actually. So it helps elucidate a bit more what these synaptic biomarkers are reflecting, and also we are interested in knowing whether there were differences between what each of these synaptic biomarkers mean or reflect in the brain. So this might have implications if it's a bit further studied and maybe validated in other cohorts, might have implications in terms of maybe using these biomarkers for clinical trials as secondary outcomes monitoring, or yeah, just to also understand a bit more the pathogenesis of the disease.